Okay, moving on to section 10.3. Now that we're going to look into DNA replication, you're going to notice that there's a couple of complications that can come about. So a few things can occur. Oops, sorry about that. There's 7 issues that must be resolved during DNA replication. First off, we have to resolve the unwinding. There's something known as reduction of increased coiling, synthesis of the primer, a lot of different things that have to be kind of fixed in order to, and like resolved in order for DNA replication to continue. Okay, so the first issue that has to be resolved for DNA synthesis to actually occur and DNA replication to occur is that the DNA needs to be separated, right? So we need to unwind the DNA helix. And this is carried out, so the 2 strands that are holding, that are, you have in your typical double-stranded DNA have to be completely separated. And this happens through enzymes that we call DNA helicases. Okay, there's a couple of enzymes that are part of the DNA helicases. includes DNA A and DNA B and DNA A, DNA B, and DNA C. There's all sorts of different things that are like different kinds of proteins, at least found in bacteria, that are part of the DNA helicases. In eukaryotes as well, we simply have helicase enzymes that unwind our DNA. And so these basically separate the strands of the 2 DNA strands from each other and unwind them, because don't forget that they are helicized. I know it's not what you see in the picture here, but DNA is helical, so and they need to be unwound. So they are they're the first proteins basically that initiate the unwinding step. They bind to the OREC. So remember that origin of replication part in the sequence. That sequence has a specific actual DNA sequences and segments to it. These are known as dimers, where they have like these 9 they're either 9 to 13 bases, specific bases that basically distinguish the origin of replication, that DNA segment, from all the other ones. They're known as either dimers or 13mers. Right, and so this is where they bind to. It requires ATP. It causes the helix to destabilize and to open up, and it exposes the single-stranded DNAs to basically the environment now. So now that we have the single-stranded DNAs, now we actually have our template strand to potentially use, right? DNA helicases can also go ahead and like recruit the holoenzyme poly3, which is the enzyme we just looked over in the prior section. And of course, they require energy in order for this to happen, because they're breaking down hydrogen bonds between the base pairs, right? They're completely separating it. It is, in order for them to destabilize the DNA structure, it requires energy. So now that we've destabilized our DNA structure and we have something like this going on, right? So our DNA, part of it is still double-stranded, the other part is single-stranded. In order for these 2 strands, they're complementary to each other. In order for these 2 strands not to bind back together, we have proteins known as single-strand binding proteins that kind of bind to this single-strand DNA and stabilize it and basically prevent it from binding back to each other. So this is what we call SSBPs, right? Single-stranded binding proteins. They bind specifically to the single strands of DNA, and they stabilize the open conformation of the helix. The other problem that results is when you are obviously opening up, especially with circular DNA, right? So if this is what the DNA looks like, it's helical, and then you're obviously opening it up in the middle to create that replication bubble. Now we have those 2 forks. Can you see how we basically have this extra coiling and this extra tension that occurs on the sides of the DNA, on both sides? This is not good. This is fine for my headphones, right? Actually, it's probably still not good for my headphones. It's especially not good for DNA. We don't want it to be tightly coiled and just like tense. We call this supercoiling. And so we have specific enzymes of the family called topoisomerases. These enzymes basically, like, they cut up the DNA, right? They cut up the DNA, cut it up, and they let it unwind. Sorry, it's a mess, but they let it unwind, and then they seal them back up. So they make single-stranded or sometimes even double-stranded cuts that basically remove the tension of the DNA from the DNA. And a big example of the type of a DNA topoisomerase that occurs in bacteria is actually known as DNA gyrase. And we even have some like antibiotics that work against DNA gyrase in order to actually kill the bacteria. Because we have topoisomerases in our own bodies as well, but they're just different. They have different kinds of proteins, different enzymes. We don't actually have DNA gyrase, which is specifically to bacteria. So it's an enzyme that relieves this coil tension from the unwinding of the helix. This is known as DNA supercoiling, and this requires ATP. Okay, so some of the genes and then also obviously their respective proteins that they encode. Some of them, these are just a list of the genes that are involved in DNA replication and their respective proteins. So we talked about DNA polymerase 1, right? That's the one that removes the primer and then fills in the gap. DNA polymerase 2, mainly necessary for DNA repair. DNA polymerase 3, that's the main synthesizing one, the one that basically makes and elongates and synthesizes and polymerizes the DNA. Primase is the one that makes a primer. We have initiation like DNA A, which is a type of helicase, and then DNA B, which is the main helicases that we need to open up the ORC. DNA A also kind of like starts recruiting everything next to it, like near it. We have gyrases, DNA ligase, which we'll talk about in just a second, and then another helicase, and of course your single-stranded binding protein. So these are just some, right? These are pretty much all of the ones that we need. These are known as the replisome. All of the enzymes that are necessary to allow for DNA replication to occur are called the replisome. So let's talk more about the primases, because this is a problem, right? The problem is, another problem that we have to basically encounter and solve in DNA replication is that the DNA polymerase 3 or any other kind of DNA polymerase require a little bit, remember how I said they require some nucleic acid to actually work on, they require a free 3' OH end. So they require something to work on to elongate. And so we need a primer to be made. This primer is made by primase, which is actually an RNA polymerase. RNA polymerase because it polymerizes RNA. And if you remember, the primer is a small short segment, about 10 to 12 base pairs long of RNA, right? And so once we have this additional primer, then our DNA can elongate in the 3' to 5', in the 3' direction. So 5' to 3' direction. So this is my primer right here, and then we elongate using DNA polymerase 3. Okay, these primers are then excised out or like cut out by DNA polymerase 1, and then their gap is filled in by DNA polymerase 1. So this brings us to the next problem that we have to solve in DNA replication. That's the continuous versus discontinuous DNA, right? So you remember that the two strands of double-strand DNA are anti-parallel. One of them goes from 5' to 3'. The other one's anti-parallel, goes the opposite direction, 3' to 5'. No, 5' to 3', 5' to 3', like that. So I'm actually going to change the video again so that you look at my thing, at my paper. Okay. So as you see, right, we always have DNA looks like so, 5' to 3', and then the other one is the opposite. Okay, when we're elongating the DNA, this is a short segment, but you usually have to deal with really long segments of DNA, right? And I'm going to try and make a replication bubble. I know this one's not really up, but like imagine that this is a replication bubble. So this is where our ORI, or the origin of replication, is happening. So this is 5' to 3', 3' to 5'. So this is our replication forks, right? This is bidirectional. These are going to continuously open up as DNA gets added on and as the DNA gets synthesized. So when we first have our primers, right, say we have our primer gets made here, right? Our primer gets made here by primase, and then now we have this 3' end, and this is a 5' end. So the DNA polymerase can now attach to this one right here and elongate that way, right? So it elongates, it synthesizes the DNA according to what complementary bases we have, and it goes until this way. And so for this strand, it's fine because the replication bubble, the fork, is just going to continue to expand, and this can just continuously go all the way down as this kind of expands and gets deeper into the DNA. This DNA polymerase can just follow it all the way down, okay? So this This strand right here is known as goes through continuous replication. And so we call this the leading strand, okay, because we have continual replication. So there's an actual definition here on the screen for you guys as soon as I get to the screen. So along one template strand of the DNA polymerase 3 synthesizes a leading strand continuously, okay, moving towards the replication fork. So this right here is going to be your leading strand. It's moving towards the replication fork. It basically follows, like as the replication fork opens, it's following that replication fork. So we have continuous DNA synthesis, all right? However, for the next strand, you'll notice this, for the next strand, so here, okay, your RNA primer, the primase is going to be made here. I'm using another color, sorry. So the primer is going to be made here, okay? If this is my 3' strand, this is my 3' end on this bottom strand, that means my 3' end is going to be right, oh, sorry. This is my 3' end. My 3' end is going to be right here. Do you see that? Yeah, so my 3' end is going to be right here. And so again, my DNA polymerase is going to come up here and it's going to extend that way. And it's going to continue to extend that way, no problem. But as you can see, as the replication bubble or the replication fork opens up, now we have this empty spot. This spot right here doesn't have any, like it's not being replicated. And actually, in fact, as the replication fork kind of opens up, this entire bottom strand right here doesn't get replicated because our DNA polymerase went towards the right. It didn't go towards the left because it's going to the 3' direction. So what ends up happening is that once this opens up, another RNA primer, as soon as we have enough opening, another RNA primer has to be made by primase. And then, so another 3' end is here. And then DNA polymerase can continue and extend it until it gets to this other segment that it just made. Do you see that? So this is known as discontinuous, okay? This is known as discontinuous because it's just not one long continuous DNA replication. So here, this is known as a lagging strand, okay? So this is called the lagging strand. And we have discontinuous DNA synthesis moving towards the replication fork. Maybe this picture here, you'll notice it a little bit better. Not really. Here we go. So the lagging strand for this replication fork right here, the leading strand goes toward, this is opening, right? So the leading strand just continuously synthesizes the DNA as the replication fork opens. And in this case, the lagging strand, it has to wait for the replication fork to open a little bit before it can basically put the primer and then extend towards the opposite direction of that replication fork. And then it's going to have to open a little bit more to put another primer and then extend in the opposite direction. So the DNA polymerase 3 in the lagging strand works in the direction away from the replication fork. And we start getting all of these little segments, lots and lots of little segments, each with a little primer. And we call these segments Okazaki fragments because it's from the guy who actually discovered, it's actually wife and husband. Anyways, the guy who discovered the people who discovered these fragments in the first place. So these fragments tend to be 1,000 to 2,000 bases long. They're even shorter in eukaryotes. So remember, we're talking about bacteria here. And we just have all of these segments that are just discontinuous, right? And so each of these segments is primed separately. And so we obviously have to change it. And you can see the lagging strand, right? There's leading strand, just follows the replication fork, leading strand follows the replication fork. Lagging strand has to wait for the replication fork to open before it can prime it and then extend it towards the 3' direction. Open up a little bit more, prime it, and then extend it. Yeah. Okay. So after the Okazaki fragments are made, you then need another enzyme, remember DNA polymerase 1, to go ahead and remove these RNA primers. So these RNA primers, these orange pieces, are removed because we don't want part RNA and part DNA. We just want full 100% DNA. They're removed, and then RNA primer kind of like fills them in. But as you can see, there's still little gaps. Even though DNA polymerase 1 goes ahead and cuts off the RNA primers from each Okazaki fragment and fills it in. So anywhere you see these orange colors, now they become blue. But even if everything is blue, you can see that these segments are still not completely bound to each other. And this is where we get DNA ligase. This is where DNA ligase is really important, because DNA ligation, it ligates or joins the DNA fragments together. And so we have a smooth, continuous, eventually one smooth and synthesized DNA molecule. And you may think that the lagging strand, also it's in the name, you may think that the lagging strand kind of falls behind the leading strand, and it does not occur, it doesn't occur simultaneously. And you actually, even though the name kind of suggests it, that's actually not what happens. There's proof to say that they do occur simultaneously. Synthesis of both strands occurs simultaneously and at the same rate. Yeah, it doesn't really get delayed. This is because the lagging strand, in order for this to happen, for them to happen concurrently, the lagging strand goes ahead and forms a little bit of a loop, right? This loop, and so DNA polymerase can work at the same time with the leading strand and lagging strand. It does lag behind just a teensy bit, but just by milliseconds, not even milliseconds, like whatever's less than milliseconds, nanoseconds, very small amount. And so they basically work together simultaneously because the lagging strand is looped, right? So this kind of inverts the physical, but not the biochemical direction. And then your fancy DNA polymerase 3 can just kind of work together. And so like work on both the leading strand and the lagging strand. If you guys remember, there's at least 2 core enzymes. Each of those, one of the enzymes is going to work on the leading strand. The other core enzyme is going to work on the lagging strand. So there's at least 2. And so this is the reason why it's possible, because the lagging strand loops in. So we basically increase that processivity, that ability for the DNA polymerase to stay attached to its template for a really long time. So to understand a little bit more about this lagging and leading strand scenario, there's an animation for you guys, right? So I'm not going to play it here on this video, but definitely go and see it, and the PowerPoint's going to be right there for you guys. The other thing that we have to talk about is the ability for DNA polymerase to proofread. So remember how I said that DNA polymerase can basically go back and recognize the fact that it's made its errors, right? And then excise, like go have exonuclease activity, kind of reverse direction, go in the 5' direction and eat away at the nucleotides. And one of those nucleotides is going to have that error, that erroneous, like bad nucleotide that it just added in. And then your normal synthesis in the 3' direction is going to happen, and hopefully at that point they'd actually add and synthesize the correct nucleotide. So it's not a perfect process, DNA synthesis. Sometimes errors do occur, but when these errors do occur, for the most part, because DNA polymerase has proofreading activity by the epsilon subunit, it can go back and get rid of it. So this is all known as proofreading. It excises those nucleotides and those bad nucleotides and fills them back up with the correct one. So this increases the fidelity of the polymerase by 100 times. And this basically severely decreases the amount of replication errors we have. So and this is really big. If we didn't have this proofreading ability, our errors would be off the charts. Just too many, way too many errors, right? So this is section 10.4, just a coherent model summarizing DNA replication. All of these, remember, all of these enzymes that we talked about are all part of the replisome. We need helicases to unwind. We need single-stranded binding proteins to keep those unwinded single-stranded DNAs from coming together, from joining back together, so they stabilize them. We need topoisomerases like DNA gyrases in the case of bacteria to basically decrease and reduce that negative tension that comes from that supercoiling. Primases to make our primer, because we know that these enzymes like DNA polymerases, like DNA Pol 3 or whatever, or even DNA Pol 1, require a free 3' OHN. So they require a small little primer, right? Small chunk of nucleic acid to work on. That nucleic acid is RNA, because primase is an RNA polymerase. So RNA polymerase is able to basically make a little primer from scratch. It doesn't require any free end or anything like that. It just reads the DNA template and starts making nucleotides. And then DNA polymerase can then attach to this really short 10 to 12 base pair primer, attach to it and start elongating and synthesizing. Here, the synthesizing could be synthesize a leading strand if it's continuous, or the lagging strand if it's discontinuous. And if we have the lagging strand, we're going to start making these Okazaki fragments, right? these Okazaki fragments. And here, every single fragment, even the one on the leading strand that has an RNA primer, at this point, the DNA polymerase 1 is going to come out and basically clip off those primers, take them away, and substitute it, like fill in the gap with DNA, right? But now we have to, especially for the Okazaki fragments, we have to actually put them together, and that's carried out by DNA ligase. So that's kind of bacterial replication in a nutshell. Very important to remember, definitely important to remember. So I believe that's it. Yep. So that's it for section 10.4, and we'll continue to section 10.3 and 10.4, and we'll continue with the next section in another video.